This is the second podcast for the digestive tract. In this image, identify villi, crypts of lubricum, simple columnar epithelium with darkly stained goblet cells, lamina propria, muscularis mucosa, and the submucosa with characteristic submucosal glands or Brunner's glands, and also the muscularis externa. The presence of these submucosal glands would make this image one of the duodenum. I'd also like to make a comment. The duodenum being a portion of the small intestine, you can notice the villi that project into the lumen of the small intestine. This is a major distinguishing feature from the small intestine as compared to the large intestine, which you'll see a little bit later in this podcast. Another view of the duodenum, you can identify the villi, the crypts of lubricum, simple columnar epithelium, the lamina propria, muscularis mucosae, and submucosa with Brunner's glands, the inner, circular, and outer longitudinal layers of the muscularis externa, and the serosa. Note that on this slide the mesothelium of the serosa is not visible. Here's a profile of the jejunum. In the jejunum you can also identify villi, crypts of lubricum, the simple columnar epithelium with goblet cells, lamina propria, the inner circular and outer longitudinal layers of the muscularis externa, and the serosa. The muscularis mucosae and submucosa are not as readily apparent at this magnification. I do want to make a comment. When you look at the various regions of the small intestine, the duodenum, which you can see on this slide, has the submucosal glands. You lose the submucosal glands in the jejunum and ileum, so the presence of the submucosal glands becomes a major distinguishing feature for which portion of the small intestine you happen to be looking at. Here's a profile of the ileum in the small intestine. You can identify villi, crypts of lubricum, simple columnar epithelium with goblet cells, the lamina propria, muscularis mucosae, the submucosa, and the inner, circular, and outer longitudinal layers of the muscularis externa. You can see the villi projecting into the lumen in this profile of the ileum, and you can also see the larger plica circularis, which is an infolding of the mucosa and submucosa in the small intestine. The plica are analogous to the rugae that you find in the stomach. Now, you can barely see at this low magnification some diffuse lymphoid tissue, some evidence of some lymphatic vessels and lacteals. Lacteals are essentially large lymphatic capillaries that are found within the villi in the small intestine, and lacteals are usually fairly well distended in this type of preparation. They are often very well distended after an individual takes a large fatty meal. The lacteals function to transport chylomicrons that are absorbed in the intestine by the lymphatic circulatory system to the liver, where the lipids are metabolized. This is a higher power view of the simple columnar epithelium in an intestinal villus. You can see a few goblet cells in the slide. You can locate the brush border, and you can see the lamina propria. This image is like the very tip of a villus. Here is an enlarged view of the crypts of lubricum in the small intestine. You can identify the simple columnar epithelium, which contain Paneth cells. The large granules of the Paneth cells are unstained in this plastic section. Oftentimes they're eosinophilic in a typical H&E stained preparation. They can also stain in other colors depending on the type of stain you use. Paneth cells are actually a type of cell that secretes lysozyme. It's an antibacterial substance. And here's a view of the small intestine that's stained with iron to particularly demonstrate the Paneth cells, the dark staining cells that are at the base of the crypts. And here you can see a higher power view of the Paneth cells at the base of the crypts. You can identify the apical granules of the Paneth cells. In this enlarged view of the crypts of lubricum in the small intestine, you can identify simple columnar epithelial cells, you can identify goblet cells, and you can identify numerous mitotic figures. Mitotic figures would be fairly common in the cells at the bases of the crypts of lubricum. This is a small intestine. I don't remember if it's from the jejunum or ileum, but you can identify on the extreme right part of the slide crypts of lubricum and lamina propria, and then you can identify the muscularis mucosae. Below the muscularis mucosae, you can identify the submucosa and the inner circular layer of smooth muscle in the muscularis externa. In the submucosa, a portion of a submucosal plexus is visible at the thick arrow. The submucosal plexus is also known as a Meissner's plexus. Here's another view again of the small intestine, either the ileum or jejunum. You can identify crypts of lubricum at the top of the slide, the lamina propria. Muscularis mucosae is nicely defined here. You can also identify the submucosa and the inner circular and outer longitudinal layers of the muscularis externa. A serosa is present here, but it is very thin, and it's only identified by the nuclei that are on the very outside surface. The thin arrow in the submucosa highlights a submucosal of Meissner's plexus, and the thick arrow between the two layers of the muscularis externa highlights a myenteric plexus, also known as an Auerbach's plexus. Recall that neurons in these plexes and neurons that are found elsewhere in the body have very large nuclei with prominent nucleoli. This is a much higher power view of an area like we saw in the previous slide for the myenteric plexus. You can clearly see the neurons with the very large nuclei and nucleoli. These plexes are between the inner circular and outer longitudinal layers of the muscularis externa. You can also observe rather nicely on this slide the serosa. Here's a section of the small intestine that's been stained with the periodic acid Schiff reaction, or PAS reaction, and this is to highlight carbohydrates. You can clearly see the goblet cells that stain very predominantly with PAS, and you can see the glycocalyx, which would be adsorbed onto the brush border of the epithelial cells in the small intestine. Here we can identify the Muscularis Mucosae running both circularly and longitudinally in this section. You can, at the very bottom of the slide, identify the bases of some crypts of lubricum. Above the Muscularis Mucosae, you can see a region of the submucosa. Note the venules that you can see within the submucosa. I particularly like this slide because it clearly demonstrates that the Muscularis Mucosae has two layers of smooth muscle, an inner circular and an outer longitudinal layer. This is a diagram of the appendix. In the appendix, you can note that there's a simple tubular glands with the crypts of lubricum. This would be part of the mucosa. You can note the fairly close association between the epithelial lining and the very extensive nodular lymphatic tissue that you'd find in the appendix. One thing to keep in mind, oftentimes the lumen of the appendix can appear very angular because there's such a large amount of lymphatic tissue that's in the submucosa. This is a view of part of the mucosa of the appendix. We haven't looked at the colon yet, but you'll note when we look at the colon that the mucosa of the appendix and of the colon look almost the same. On this particular slide, you can note the fairly large lymphatic nodule and lots of diffuse lymphoid tissue. This lymphatic nodule is probably broken through the Muscularis Mucosae, so much of this nodule is actually within the lamina propria. Here's a profile of the large intestine, and a couple of points I want to make on the slide. First, you can see the mucosa. Note that the glands in the mucosa are simple straight glands. Note that there are no villi projecting into the lumen of the large intestine as you would have had in the small intestine. You can just barely see some profiles of the Muscularis Mucosae. There's a huge lymphatic nodule on the slide, on the right part of the slide, that may be disrupting the Muscularis Mucosae. So this nodule is residing both in the submucosa and maybe pushing up into the lamina propria. There's a lot of diffuse lymphoid tissue. You can see some blood vessels and perhaps lymphatic vessels in the submucosa, but I want you to pay particular attention to the Muscularis externa and note the outer layer of the Muscularis externa, how much exaggerated it is. The muscle bunches up into a tinea coli. You may recall in gross anatomy when you dissected the abdominal cavity and you looked at the large intestine, you could see three bands that were running on the outside of the large intestine. Those three bands would be the tinea coli. They're just an accumulation of the outer layer of the Muscularis externa. Here you can identify in the large intestine the fairly straight glands, the crypts of Libricum, typical of the mucosa of the large intestine. You see lots and lots of goblet cells. On a histological section, you almost think that it's exclusively goblet cells. That's not really true. The goblet cells may only comprise 20 or 30% of the epithelial cells here, but nevertheless, because there are so many, it just appears that there's a preponderance of goblet cells. You can make the general point that as you move down the intestine, the number of goblet cells does increase. You can note the lamina propria is fairly heavily infiltrated with lymphocytes. You can see the Muscularis Mucosae on the bottom of the field, and just a little bit of the edge of the submucosa would be present below the Muscularis Mucosae. Here's a profile and cross-section. The base of the glands are at the crypts of Libricum, so you can see the glands themselves in cross-section. You can identify lamina propria and some capillaries in this section. And this section would actually be cut fairly deep, like I said, at the level of the crypts of the glands, but it would be running parallel to the surface of the organ. Here is a view through the anal canal. This section is probably through the pectinate line, would be visible in gross dissections. Here you can note a transition of epithelium from simple columnar epithelium with goblet cells to non-keratinized stratified squamous epithelium. You can observe the intermingling of the two types of epithelium at various points here on the slide, and this is just simply because the tissue has folded in on itself as it was prepared. You can identify the lamina propria and the Muscularis Mucosae. I'd like you to think about some transitions here. Imagine you were endoscoping the patient and moving from the outside into the colon. You would see in the rectum that would be a stratified squamous with keratin to the anal canal, which would just be a stratified squamous, which you see here on the left of the slide, and then you get an abrupt change to the simple columnar epithelium that you'd find within the colon. So I'd like you to keep that orientation in your mind as you think about the arrangement at this portion of the large intestine.